Hey Team Bio, welcome to your second semester screencast. So for those of you who are new to this format, a screencast is a little video lecture that I record outside of class. You watch it for homework and then you come on into class the next day. You take an open note, open partner quiz on the content of the screencast, and then we do something with it in class. Part of the point of doing these screencasts is so that I can minimize the time I spend lecturing you in class, and we can spend more time experimenting, building, tinkering, whatever we're observing, playing with microscopes, doing whatever we're going to do in bio class that you really can't do at home. Okay, so let's get right to it. You should treat this like a lecture that I would give you in class, where you listen along, you take notes, and you write down some questions. There'll always be a time to ask your questions about the screencast at the beginning of class the next day. Okay, so here we go. Proteins, or we're going to talk about energy coordinate diagrams today. We are moving on to our third of the big four. So we've talked about carbohydrates, we've talked about fats and lipids, and now we are talking about proteins. Specifically, we're going to be talking about enzymes, which are a subcategory of proteins. Enzymes are all proteins, but not all proteins are enzymes. So for example, proteins can play a role as hormones in your body or structural functions in your body. So enzymes are just one small class of proteins. Okay, well, you all know proteins. What are they made of? Their monomers are amino acids. There are 20 different types of amino acids. 8 of them are essential, meaning you have to eat 8 of them. These are the monomers. When you take a bunch of amino acids and you string them together, you get a polypeptide. This is the polymer. But in order to actually get a functional protein, you need to fold the polypeptide in a specific manner to get a protein. So the shape of the protein is really integral to its function. Enzymes are folded in very specific ways to create specific patterns and shapes. And we will have started building a protein in class when you watch this, and we will continue to build a very small protein in class over the next couple days. Okay, enzymes. They're one subclass of proteins. They are biological catalysts. A catalyst is something that speeds up the rate of the reaction. By lowering the activation energy. And a catalyst is not consumed. In a reaction. So if you have a catalyst present and you have all of the inputs or the reactants to a reaction, the catalyst can keep working, doing its job again and again and again until all of the reactants have been converted to products. So these enzymes, in order to do their job, they need to be folded in a specific shape. So in order to be biological catalysts, we need to fold them. Okay, there are 2 major types of reactions in this world. They're called exothermic reactions and endothermic reactions. Exothermic reactions are reactions that release energy. When we look at the overall energy of the inputs, the reactants, compared to the energy of the products, we see that the reactants have higher energy than the products. So the difference between the energy of the reactants and the products is called delta H. And we see here that delta H is negative. The reactants have a higher energy than the products. So this amount of energy will be released in the reaction. In an endothermic reaction, this type of reaction requires energy. So when we look at the energy of the reactants, and we compare it to the energy of the products, we see that the difference between them, the energy of the reactants compared to the energy of the products here, I don't know why this doesn't want to make a line for me. This again is delta H, and it is positive in an endothermic reaction. We need to put energy into this reaction in order to add energy to the reactants to turn them into the products. So this amount of energy is required in order for the reaction to occur. Two major classes of reactions, exothermic and endothermic, and two really important reactions fall under those two categories. So cellular respiration and photosynthesis are the two big cellular processes that we're going to spend a pretty good chunk of time in the winter talking about. Cellular respiration is the way that we take glucose, the energy of glucose, and we convert it into usable cellular energy, ATP. And you'll see here that the reactants, so glucose and oxygen, have higher energy than the products, which are carbon dioxide, water, and some energy is released in this process. So when you eat food, almost all this food is going to be processed in cellular respiration, and you are going to exhale CO2 and water and release energy as well through cellular respiration. So this is an example of an exothermic reaction. Photosynthesis is an example of an endothermic reaction. If you look here, we have the same set of reactants and products in cellular respiration as we do in photosynthesis, except the reactants of photosynthesis, or sorry, the products of photosynthesis are the reactants of, the products of cellular respiration are the reactants of photosynthesis, and the reactants of cellular respiration are the products of photosynthesis. So we start out in photosynthesis with carbon dioxide and water and some energy from the sun, and we form glucose and molecular oxygen. But this whole process requires energy in order to produce glucose and oxygen. Okay, so what does this all have to do with enzymes? Well, exothermic reactions and endothermic reactions, we both have, you'll notice here that before we go from reactants to products, so imagine we're burning a log here. You know that when you have a log and you light it on fire, it releases carbon dioxide and water. Maybe you didn't know the water part, but it does. It reacts, the glucose burns with oxygen in the air and combusts to form CO2 and water. And then the energy of that CO2 and water is much less than the initial log, which was full of glucose. And that energy is released as heat. But in order to get the log to burn, you have to actually light it on fire. It's not just going to spontaneously combust and turn into CO2 and water. This energy of getting the reaction started is called the activation energy. And if you go from the peak of this curve down to the initial energy of the reactants. This is the measuring our activation energy. It's the amount of energy required to get a reaction started. We can draw the same diagram here. I made this a little bit too long, where the difference between the peak and the final products is our activation energy. And in the case of an endothermic reaction, this is like the extra little bit of energy you need to put in order to get the products created, on top of the overall energy you need to get in to have the products form. Okay, so this black line here is showing us the rate of a reaction without an enzyme. And in blue, on both of these graphs, we are going to draw the rate of reaction with an enzyme. So with an enzyme, the initial energy of the reactants is the same as it is without an enzyme. But one of the big differences is that a reaction that has an enzyme lowers the overall energy. Oh, I don't know if it's going to let me do a curve here. It doesn't want to. Okay, I'm just going to freehand this. Lowers the overall energy required to get the reaction started. So we haven't changed the energy of the reactants. We haven't changed the final energy of the products. What's changed here is that with the addition of the enzyme, you don't need as much energy to get the reaction started. So we've lowered the overall energy of activation. So this is activation energy with an enzyme. And as a result, you'll notice here that we create the products much more quickly than without the enzyme. Because the energy of activation is lower, the reaction proceeds more quickly. Okay, and then we can draw the same thing over here. We actually start with the exact same energy of reactants as well in this diagram. We kind of follow the same curve up, but then, doesn't want to let me do a, there we go. A reaction with an enzyme is going to have a lower overall activation energy. There we go. Okay, how do enzymes do this? Well, you saw in class today that enzymes have a specific shape with an active site. And they really, the way that they catalyze their reactions is by placing their substrates or their reactions in very specific positions. Substrates for the reactants. And when they put their substrates in specific positions, then they put their reactants in specific positions, they make it much more likely for the reaction to occur. Okay, so this is all I have for you. We've got endothermic and exothermic reactions. Exothermic is going to release energy, while endothermic is going to require energy. Both endothermic and exothermic reactions go more quickly in the presence of an enzyme, and this is because an enzyme is going to lower the amount of energy that's required to get the reaction started, and they do that by placing their substrates, the reactants, in really specific positions so that it's easier for the reaction, or it's more likely that the reaction is going to occur. Okay, that's all.